Have you ever really stopped to think about how a simple pattern of light becomes, well, a conscious thought, an image, a memory, a face you recognize? Today, we are going on a deep dive, a masterclass, really, into the visual pathway. We're going to trace that incredible neural highway that transforms photons into pure perception. We'll follow the signal every step of the way, from the back of your eye right into the very core of your brain. And by the time we're done here, you'll be able to map out every single component of this system, know exactly what it does, and even understand the classic visual problems that pop up when this super intricate pathway gets damaged. So let's get right into it. So here's our game plan. First, a quick introduction to the visual system as a whole. Then we'll zoom right into the retina. That's where it all begins. From there, we'll follow the signal to a part of the brain called the thalamus, and then on to its final stop in the cortex. And to wrap it all up, we'll look at the clinical side of things. What happens when this amazing system goes wrong? All right, let's kick things off with the big picture. You know, it's easy to think of our visual system as just a camera, but it's so much more. It's really like a superhighway for information, a highly organized route that starts in the eye and travels way back into the brain. And trust me, every single stop along this highway has a job to do, and it's a critical one. This diagram here shows one of the most fundamental rules of how our brains are wired up. It's called contralateral processing. It sounds complicated, but it's pretty simple. Everything you see in your right visual field, that gets handled by the left side of your brain. And everything you see in your left visual field, yep, you guessed it, handled by the right side of your brain. This crossover is a core design feature of the visual pathway, and we're going to see exactly how the brain pulls this off in just a bit. So where does this incredible journey even begin? Well, it all starts at the very back of the eye, in this thin, delicate layer of tissue called the retina. This is where the real magic happens. This is where physical light energy gets translated into the electrical language of the brain, what we call a neural signal. Now, the retina isn't just one simple layer. It's this incredibly complex, multi-layered stack of specialized cells. What's kind of weird is that light actually has to travel through all these layers to get to the very back, where it finally hits the photoreceptors, the famous rods and cones. From there, the brand new neural signal travels forward again, through other cells, eventually reaching the ganglion cells at the front. The axons from these ganglion cells are what bundle together to leave the eye. We'll also see how a few other helper cells, like amacrine and horizontal cells, help fine-tune this signal before it ever leaves. And here they are, our two types of light-detecting cells side by side, a rod on the left and a cone on the right. You can see they follow a similar blueprint, right? They both have an outer segment that's packed with light-sensitive pigments, an inner segment that's basically the cell's power plant and factory, a nucleus, and then a synaptic terminal to pass the signal along. But, as you can see, their distinct shapes are a huge clue to their very, very different jobs. So first up, the rods. And get this, we have a staggering 130 million of these things. They are the absolute masters of night vision and picking up motion in our peripheral vision. Structurally, you've got the inner segment, a long rod fiber with the nucleus, and that all-important outer segment, which contains about 700 little discs just packed with a pigment called rhodopsin. Now here's the part that's super fascinating and completely counterintuitive. In total darkness, rods are actually active. They're constantly releasing a chemical messenger, a neurotransmitter called glutamate. But when a single photon of light hits that rhodopsin pigment, it kicks off a chain reaction that shuts off the glutamate release. The brain detects this stop in the signal, and that's how it knows light has been detected. Wild, right? Okay, next up are the cones. These are the cells that handle our sharp, detailed, high-acuity color vision. We call this trichromatic vision, and that's because we have 3 different types of cones. Each type has a slightly different pigment that's tuned to a specific wavelength of light, one for red, one for green, and one for blue. By combining the signals from these 3 types, our brain can construct every single color we perceive. And structurally, as you can see, they're a bit more compact than rods, made up of mainly an inner and an outer segment. So, the signal from those photoreceptors gets collected by ganglion cells, and these come in 2 main flavors. First, you have midget cells. They have these small, dendritic arbors. You can think of them like tiny, fine nets for catching signals. This makes them perfect for processing fine detail, smaller, slow-moving things, and of course, color. Then you have parasol cells. They have large dendritic arbors. Think big, wide nets. This makes them great for detecting large, fast-moving stimuli, and they're super sensitive to low contrast. But, and this is key, they don't process color. So right here in the retina, our visual signal is already being split into 2 parallel streams, one for detail and color, and one for motion and contrast. And these 2 streams are going to stay separate for a long way. But wait, before the signal even leaves the eye, it gets processed by a whole team of editors. You have horizontal cells, which provide inhibitory input that helps to enhance contrast, making edges appear sharper. Then there are amacrine cells, which are a bit more complex. They provide both inhibitory and excitatory inputs to shape the signal in all sorts of ways. And finally, interplexiform cells create these little feedback loops between the other two, allowing for even more fine-tuning. This is basically the retina's own little image processing chip, cleaning up the signal before it ever gets sent to the brain. Okay, so the signal has been captured, processed, and edited. Now it is time to leave the eye. All those axons from the ganglion cells, they all bundle together to form the optic nerve. This is the great data cable connecting the eye to the brain. Our next stop is a really critical intersection called the optic chiasm. We can actually break down the optic nerve's journey into 4 distinct parts. First, it starts right inside the eyeball. That's the intraocular portion. Then, it travels back through the bony eye socket, which is the intraorbital portion. Next, it has to pass through a narrow bony tunnel called the optic canal. That's the intracanalicular portion. And finally, it enters the skull itself as the intracranial portion, making a beeline for its first major junction. And here is that junction, the optic chiasm. The name actually comes from the Greek letter chi, which is shaped like an X, and that perfectly describes what it does. This is the point where some, but not all, of the nerve fibers from each eye cross over to the opposite side of the brain. So the optic chiasm sits just above a bony structure called the sella turcica, and inside that sits the pituitary gland. Now this location is clinically super important, and we'll see why in a bit. Functionally, this is where a critical sorting event happens. The axons from the nasal half of each retina, that's the half closer to your nose, they cross over or decussate to the opposite side of the brain. But the axons from the temporal half, the side closer to your temples, they stay on the same side. This elegant rewiring is the exact mechanism that makes that contralateral processing we talked about at the beginning possible. So once those fibers pass through the chiasm, that resorted bundle of nerves gets a new name, the optic tract. Now we follow it even deeper into the brain, to the great relay station known as the thalamus, before it makes its final jump to the cerebral cortex. That's where conscious vision actually happens. And here, the path splits. The main highway, carrying about 90% of the information that we need for conscious perception, goes to a very specific spot in the thalamus called the lateral geniculate nucleus, or LGN for short. The other 10% takes an older, more primitive route to areas like the superior colliculus, which handles eye movements and reflexes, and the pulvinar nucleus. And it's interesting, the pulvinar is actually the largest nucleus in the entire thalamus, but scientists are still working out its precise role in vision. Let's focus on that main pathway for now. We arrive at the LGN, and as you can see from this diagram, it's not just some simple bus station. It's an incredibly organized structure, like a 6-layered sandwich of neurons. And as we're about to see, each of those 6 layers has a very specific job. So here's how those 6 layers break down. 4 of them are called parvocellular layers, and they get their input directly from those detail and color midget cells we talked about back in the retina. The other 2 layers are the magnocellular layers, and they get their input from the motion-detecting parasol cells. I mean, this is remarkable. The LGN acts like a perfect sorting station, carefully preserving those 2 parallel streams of information that were established all the way back in the eye. Okay, from the LGN, the fibers fan out for the last leg of their journey. These are called the optic radiations, or sometimes the geniculocalcarean fibers. The dorsal and central bundles take a pretty direct path through the parietal lobe. But one group, the ventral bundle, takes this long looping detour forward and down through the temporal lobe. This specific path is called Meyer's loop, and its unique route is incredibly important for neurologists who are trying to pinpoint the location of brain damage. But at the end of the day, all these roads lead to the exact same destination, the primary visual cortex. And we've arrived. This is a classic map of the brain that's been divided into what are called Brodmann areas, based on their cellular structure. Our final destination, the primary visual cortex, or V1, is designated as Area 17. It's located right at the very back of the brain. This is the first place that raw visual data hits our consciousness. From here, the information gets passed along to the neighboring secondary areas, 18 and 19, for higher-level processing, things like recognizing faces, interpreting motion, and actually perceiving color. So we've just mapped out the entire healthy pathway. But its intricate design also means it can be damaged at many, many different points. Let's start right back at the beginning. What happens when the problem originates in the retina itself?
Well, a major mechanical issue is retinal detachment. A blow to the eye, or sometimes just a spontaneous tear, can cause the retina to peel away from the back wall of the eye. When this happens, fluid can accumulate behind it, cutting off its blood supply and leading to blindness if it's not treated urgently. But retinal problems can also be genetic, affecting the photoreceptor cells themselves. This is what causes color vision deficiency. In dichromatic vision, the gene for one of the three cone pigments, or opsins, is missing, and in the much rarer monochromatic vision, two or even all three cone opsins are absent.
Okay, so problems in the retina are one thing, but what happens when the damage occurs somewhere along the pathway itself? Damage, or what doctors call lesions, to these neural cables creates very specific, very predictable patterns of vision loss that can tell a neurologist exactly where in the brain the problem is.
First up, if you have a lesion that damages the optic nerve before it gets to the chiasm, the result is pretty straightforward: monocular blindness. As the visual field diagram shows, the world just goes completely dark in that one eye. Common causes for this could be a stroke, inflammation from multiple sclerosis, or even a really severe migraine.
Next, what if the damage is right at the optic chiasm? This often happens because of a pituitary tumor pressing upwards and squishing those crossing fibers. Now, remember which fibers cross? It's the ones from the nasal retinas, which carry information from the outer or temporal parts of our visual field. The result is something called bitemporal hemianopia, or what you might call classic tunnel vision, where you lose all your peripheral vision in both eyes.
Our third classic pattern happens if the damage occurs after the chiasm, somewhere along the optic tract, the optic radiations, or in the cortex itself. This causes homonymous humanopia. And as you can see, the patient loses the same entire half of their visual world, either the left side or the right side. This vertical midline vision loss is a very common result of a stroke that affects the posterior cerebral artery.
And this brings us to one last, really crucial question. We've seen what happens when the cables are damaged along the way, but what about when the damage is to the final destination itself, the primary visual cortex?
Well, the result is something called a scotoma. This is a very specific partial loss of vision, like a little island-like blind spot floating in an otherwise normal visual field. Because the primary visual cortex is a literal map of our vision, a small lesion there from a stroke or an injury creates a corresponding hole in what we can see. So from a single photon hitting the retina to this complex, beautiful map in the cortex, the visual pathway is truly a masterpiece of biological engineering. Its journey shows us that our brain doesn't just see the world, it actively constructs it, one neural signal at a time. Thanks for joining me on this tour.